Hello, everybody. It's Mr. Caudale. I hope you're doing well. I'm trying to hang in there. It's mid to late November. I hope you're doing well and that your family's healthy and that you're optimistic. I'm optimistic for humanity. Look, humanity can do things like resurrecting woolly mammoths. That's the hope. I would love to show you this movie, but I'm not going to record this on this platform, so we'll hopefully watch it in class. Today, we're moving on with chapter 26. We need to be able to construct these trees. It's not enough for you to have a little funny, cute mnemonic about remembering those levels of taxonomic characterization for life that Carlos Linnaeus originated with. It's enough. What you need to be able to do is build these trees and then analyze these trees. You should have some handouts that I've given you. I'll hopefully give these to you in some way. And then here's another handout that I haven't made yet, but it just has a lot of data and stuff on it. And the idea is that you have to become comfortable at navigating these trees and building these trees. So let's start with 26.3. That's all we're going to do today. This is all about using shared characters to construct these trees. Now, these are called phylogenetic trees, but we can call them cladograms as well. And these can be used to infer a phylogeny, which is just the kind of relationship of two organisms in evolutionary space and how they relate to others. So this is called cladistics, this idea of using this information to build these trees. And we're going to group organisms by common descent from a common ancestor as they've gone through time in evolution. Remember, always on these, I like to put a timeline on the bottom of these on the x-axis. In this case, it would be going from right to left. Right would be on your screen there. Right would be present day, and then we would go back in time to the left. So here's a little movie.
Have you ever organized your room and realized that you put similar or related objects near each other and completely different objects farther away? This is similar to how scientists construct a cladogram. Cladistics is a systematic method of classification which uses shared derived characters to make a cladogram. A cladogram is a branching diagram that represents the proposed phylogeny or evolutionary history of a species or group. Cladistics assumes that each group has an ancestor that other species do not share. The cladogram in our example shows how 4 very different species could develop from a common ancestor. The common ancestor would be at the bottom of the diagram. The cladogram is constructed by sequencing the order in which the derived characters evolved with respect to the outgroup. The outgroup is the species that has more ancestral characters with respect to the other organisms being compared. In this example, mollusks are the outgroup. The closeness of the clades in the diagram indicate the number of characters shared. The group that is closest to echinoderms shares the most derived characters with echinoderms and shares a more recent common ancestor than with groups farther away. Each line that breaks away from the main line is called a node. The nodes where the branches originate represent a common ancestor.
There's something that needs to be pointed out here. If you notice between the nodes, between the places where there's a grouping of organisms that kind of comes off the main line there's some information, and it's usually about a characteristic. And I'm reading from the bottom: protostome, which is a type of tissue development in an embryo, segmentation of bodies, exoskeleton, which would be a hard skeleton, and then up there at the top is a deuterostome, which just means a poop hole, essentially. But the point is that what happens is you get an origination of a certain characteristic through evolution, and then what they're saying on this is we go from bottom to top or from left to right here. What they're saying is that all the organisms past that characteristic are going to have that characteristic, but the ones in this figure below or to the left of that characteristic that we've put on that main line there will not have that characteristic, and that's what then is going to exclude them from going further through evolutionary time with these other organisms. But number two, it's going to say that they're different They never were in the world when that thing evolved, or that wasn't a part of their development. And so that's how we kind of clump organisms together. Think about a chimp and a human versus a chimp and a snail. Like, they're going to have more characteristics similar if we went on one of these lines. So let me finish this for you.
Us, echinoderms, and arthropods have 3 derived characters in common, so they share a more recent common ancestor than echinoderms and mollusks. Now you see how scientists organize species based on shared characters, similar to how you organize your pens and pencils, together, separated from your awesome collection of music.
Yes. Okay. So to me, the beginning of chapter 26.3 is kind of basic. So let's go through a couple of these definitions here. So if we're going to create a grouping, and in one of these phylogenetic trees, we can call it a monophyletic group. And what that means is that you include the common ancestor and all of its descendants. So that's a monophyletic group, or a clade. Just another term for it. Here's another way. Well, you can see that. I guess I don't see that, that we have a common ancestor and all of its descendants highlighted here. You can see the kind of the blue and then the yellowish highlighting here. Well, there's 3 different types of this grouping: monophyletic, paraphyletic, and polyphyletic. From the bottom there, I've shown the term polyphyletic, and what that it refers to are organisms. You can just say, look at this group of organisms. They're marine mammals, and nothing else on this screen is a marine mammal. It's a mammal that lives entirely in the water. Cetaceans like a porpoise or a seal could be called marine mammals. They live entirely in the water. But if you look at their development through mammalian evolution, they are grouped together in a polyphyletic group, which means if you just take those guys in the yellow highlight, what you're not showing are any of their common ancestors together. So, and in fact, they don't even have similar ancestry if you start to look way over on the right. Yes, I get it, they have a common ancestor way to the left in this phylogenetic tree. But if you start going to the right, and some other branches have even occurred in order to get there, they're not really in the same group. You can see that there. That's called a polyphyletic grouping of organisms. We just kind of randomly grabbed these 2 for a reason. We wanted to, so it wasn't random. Sorry. Up there we have a paraphyletic group. If you wanted to say, oh, look at the common ancestor of even-toed ungulates, even 2 or 4 toed, and then ungulates are these things that I always think of an ungulate as like an elk or a deer, something that's out in nature with these. I forgot what the term ungulate means, but it has something to do with the toes. The point is that if you said, well, then porpoises are in that group, but we're going to exclude them for now. We're just going to look at the deer and the hippos here. That would be called a paraphyletic group, meaning we're going to look at these guys. We've grouped them together, but we're not incorporating everyone that is a descendant from the common ancestor that the organisms in our group actually have. So compared to its ancestor, an organism has both ancestral and derived characteristics. It's the next page in 26.3. Go ahead and take a look, press pause here. What would be some ancestral and derived characteristics for the organisms in this phylogenetic tree? Well, a shared ancestral characteristic is one that originated in an ancestor of the taxon. So way over there on the far left side of this phylogenetic tree, you could say, oh, that common ancestor for all these guys over there on the right must have had 2 eyes, or a vertebral column, or legs. Pretty basic stuff, right? That's an ancestral character that the ancestor had that now they all have as they've come through time. Well, a derived character is one that in a certain portion of the evolutionary tree, some of the organisms evolved to have. Like, for instance, down at the bottom there, the wolves maybe have pointed ears, you could say, or the badger and the otter might be smaller body size or shorter legs, or I don't know. Those are shared characters that have been derived through time through evolution. Well, this is a picture from your textbook. And what I would like you to do is I would like you to use this. And if you were in class, I would give you a blank piece of paper and I would have you draw a phylogenetic tree based on this information. So I'm looking in the 10th edition. This is on page 554 and it says draw it in the textbook. So press pause and try to draw this. How would you draw this? I mean it. Spend a few minutes on this, please. Well, what we see there is if you start at the top, you'll see that the lancelet, which is this kind of weird worm-looking thing that lives in the water, is going to be different than all the other things that are kind of downstream evolutionarily from it. It doesn't seem to have any of the characteristics that are in the y-axis here, the vertebral column, the hinged jaw, the four walking legs, the amnion. That's like an amniotic sac. That's like the sac, the white of an egg. That's basically the sac that has the cushiony, watery solution that helps mama's baby develop in her belly, or hair they don't have. And so they're going to be the outgroup. And you always have to have an outgroup. It's like a control. So you're saying there is an organism that doesn't have any of this stuff. And then what you're going to do as you cruise down either kind of the cladogram, you could draw it like this, like we saw in that movie, or you could draw it. I'm not going to give it away. You could draw it a different way. And the idea is as you go in time, whatever your axis of time is, you're branching stuff off that has more of the characteristics than the previous or ancestral organisms did. So I'm going to go there. And here's where I'm going to go. On page 54 at the bottom and in your handout, isn't it so convenient? Right next to this thing, they have the answer. The idea is what I want you to see is that as we go through time from left to right here on our axis, from back in time, x-axis to present day, you're going to see that the organisms that branch off are the ones that don't have the next characteristic. Make sure that you really understand this. Well, here's some more rules here that I got this from an AP conference. Angled and square branches mean the same thing, so you can draw it either way is fine, or if you see it on an exam, either way they're exactly the same thing. Because you can kind of see that they kind of look the same, they're just kind of angled or squared branches. Yeah. You can also rotate. They can be drawn in any orientation. You see how you can rotate the whole thing around. It doesn't matter which way it's oriented. You can also rotate around a node like a mobile. So if that's like a hanging thing that maybe the wind would catch and move stuff around. So you can take two organisms on a node or a hinge point and then flip them, and that doesn't mean anything. And you can see that here. Also, the space between the tips and branches has no meaning. You can't say, oh, that's equal to this or time or something like that. I like it, what I've always contended in this instance, if you go from the bottom to the top on the y-axis in this picture, that you could say that that's time, but not in terms of the width in this instance. Well, there's two more concepts that we need to go over in this 26.3. The principles of maximum parsimony and maximum likelihood. I have the definitions up here. If you'd like to pause it, you can. So the idea is if we had a data set of 50 organisms, and the question is, how can we place them in an evolutionary relatedness tree like a cladogram? I think I read in the book that it's like 8 times 10 to the 76th power options of how you could draw that. So it's crazy. Like, how do you draw it? How do you know that a chimp is more closely related to a human, and therefore we're going to place it on a phylogenetic tree versus a bacteria or a snail? And there are these two kind of rules that we like to use in order to build appropriate evolutionary trees or phylogenetic trees. So the principle of maximum parsimony says it's the tree that requires the fewest evolutionary events or appearances of shared derived characters that is the most likely. this comes from the work of this theory comes from the work of this guy William of Ockham, and he was a philosopher in England, I think 1033, and basically he just said, keep it simple, stupid. He said, when it comes to nature, just make the fewest assumptions possible. I remember my daughter when she was young, she got a balloon once and it was in the living room, and the next morning it was on the other side of the living room. And I said, how do you think it got over there? And she said, oh, maybe there was a clown on a unicycle and came in the middle of the night and dragged it, blah, blah. And then I thought to my scientific boring self that, hey, maybe the heater just came on and blew it over there, or something like a wind current. And the point is often, while it's boring, often the least amount of kind of assumptions that you make is often the correct answer. And you'll find that this applies to almost every scientific and mathematical concept, as well as social situations. Keep it simple. And the simpler people are, the happier they are. I just find that. But that's boring, old people stuff. I get it. The principle of maximum likelihood states that given certain rules about how DNA changes over time, a tree can be found that reflects the most likely sequence of evolutionary events. The idea is we know that through DNA replication, through the S phase of interphase, and then in preparation for cell division, and then when we know that DNA kind of mutates at a constant rate. And so we can also use that to our advantage when trying to figure out which tree is most likely. If we have competing evolutionary trees, we can say, well, we know that DNA mutates at a certain rate. This is probably the most likely one as far as evolutionary change is concerned. Here's an example from the previous textbook. If we compare a human, a mushroom, and a tulip as far as percent DNA differences between the sequences, so we say that human and mushrooms are 30% difference in their DNA, and a human and a tulip is 40% difference in their DNA. We can then look at these two different potential phylogenetic trees for placing evolutionary relatedness to humans and to mushrooms and to tulips. And off the top of your head, which one do you think is more correct? I'm going to tell you, these are equally parsimonious trees in this instance because they're assuming the same number of evolutionary events, just a couple evolutionary events which have brought these different lines of evolution here. But if we look at if we know that DNA mutates at a fairly constant rate, then we have unequal x-axes here between the two. Well, I guess specifically with the right-hand tree, it's less likely because there's unequal rates of DNA or evolution change over time. I like the tree that's on the left, and I do know, just being a biologist, that we are more closely related to mushrooms than we are to plants. I wish it wasn't that way, but it is that way. So I know that intuitively that's going to be correct, but then I just have to imagine, it just makes more sense with the numbers if you look at the percent DNA differences between mushrooms and tulips, or tulips and humans, or over time. Well, on your handout, you have this picture. Go ahead and press pause, and let's try to answer this question. Well, the answer here is A. It just assumes the fewest evolutionary events when you look at the sites in the DNA sequence and how they're different between species 1 through 4. Also, I've given you this. This is from the College Board. I'll go ahead and press pause and try to answer this, and then here are your potential answers. Well, just by looking at this, you can tell when it comes to rRNA subunit nucleotide differences. I'm walking up to point at the screen. You can tell that species 1 and 2 are not that different in terms of nucleotide differences, and species 3 and 4 seem to be pretty similar in terms of their difference from species 1 and in relatedness to themselves. So probably when I look at these phylogenetic trees, I'm going to probably clump 1 and 2 together, 3 and 4 together, and then 1 versus 5 or anything else versus 5 is going to be way different. 5 is kind of off by itself. It has way more nucleotide differences. And we know that the nucleotides then, I know this is rRNA, but in part are going to create what the characteristics are of that organism. So let's take a look here. So which one of these phylogenetic trees do you think is correct? I would say it's not going to be D. It's not going to be A for sure. It's not going to be B. So even by default, you would say C might be the correct one. 1 and 2 are grouped together, 3 and 4 are grouped together, and then 5's off by itself. This is from the College Board. Here's another question from the College Board. Here's a bunch of animals, and here's a bunch of characteristics that these animals may or may not have. We've tried to create a phylogenetic tree or evolutionary tree based on these characteristics. Go ahead and press pause and try to answer this question. I think this is kind of a dumb question. It's from the College Board, but I believe that B is the correct answer. Salamanders should not show claws or nails in the data table, and that's true because it looks like claws and nails originated in organisms after salamanders branched off of this line of evolution. Well, here's our last thing that we're going to do. I say use the handout here because I used to give this on a handout. It is in your book on page 557. You can see these 3 different birds, and you can read this. Try to place them in the correct phylogenetic tree. Go ahead and press pause. Well, the answer here is this first phylogenetic tree, that 1 and 2 are more closely related to one another. Now, maybe you could just see that just by this picture when you look at the beak and the eye and how the feathers are and the markings that the organisms have. You could do that. Another way that you could do that is to analyze their DNA, and you can look and see. So this is, I just read this before I started recording, and I understand what they're doing with their explanations on page 557 in the 10th edition, but I think an easiest way to do this would just be to look at species 1 at 4 sites on the genome, going from left to right, and you say, who is most closely related to who with respect to these sites on the genome? And species 1 and 2 look to have 2 differences as you go from left to right here, sites 1 through 4. And species 1 and 3 have 3 differences out of 4. And species 2 and 3 have 3 differences out of 4. So therefore, I would put 1 and 2 closer together in a phylogenetic tree. And that, again, that goes back to the most left-hand phylogenetic tree, or the first left column, where 1 and 2 are next to one another, and then 3. You can go through all this stuff. That's basically all they're doing there. Hey, we're done with 26.3. Thanks for your attention. I hope it helped.